Hola, ¿cómo están? Pero muy buenos días, buenas tardes o buenas noches, depende de donde nos estén mirando. Damos comienzo a un nuevo encuentro de la Sociedad Argentina de Biología, en esta sana costumbre que tenemos frecuentemente de acercarle a ustedes contenidos científicos de estricta actualidad médica y del ámbito también de la salud, claro. Algunas aclaraciones antes de comenzar. Todos nuestros encuentros, todas nuestras citas quedan grabadas en nuestras redes sociales para que ustedes puedan volver a ver, puedan aconsejar a quien lo consideren. Y claro, como también hacemos siempre, al finalizar la exposición de nuestros invitados, se va a abrir un espacio de preguntas que ustedes, si lo desean, y ojalá así ocurra, pueden realizar a través de las propias redes sociales donde están mirando esta cita. Sin más prólogo, vamos a presentar primero al doctor Juan Sabaté. Él es médico e investigador del CONICET, además es docente de inmunología en la Facultad de Medicina de la UBA. Estimado Juan, ponemos en tus manos la coordinación de este evento y también vamos a ingresar a la pantalla desde nuestra cercana San Antonio, la tierra de Manu Ginóbili, a el expositor del día de hoy. Gracias, Juan. Thank you very much. Sunil, comenzamos nomás.
Bueno, muchas gracias, Diego, y a la gente de la SAB, de la Sociedad Argentina de Biología, a Belén especialmente, por invitarme a este seminario. So I'm going to introduce you to Sunil Ahuja. Sunil is going to talk today about a very interesting and provocative topic about immune resilience. Sunil Ahuja is professor of medicine, microbiology, immunology, and molecular genetics at the University of Texas in San Antonio. He's director of the Center for Personalized Medicine in the South Texas Veterans Healthcare System, also in San Antonio. Dr. Ahuja is an expert in virology, immunology, and translational research related to infections and inflammatory diseases like HIV/AIDS, allergy, autoimmunity, and aging. He has received many honors and awards, including the NIH Merit Award, the Doris Duke Distinguished Clinical Scientist Award, and the Elizabeth Glaser Pediatrics AIDS Scientist Award, just to mention a few of them. Dr. Ahuja has made seminal contributions in the field of immunology and microbiology and medicine. Studies from his group have been published in top journals like the New England Journal of Medicine, Science, Nature Medicine, and Nature Immunology, between others. And now his lab is focused on the characterization of host determinants of immunological health. And I think today he's going to talk about this very interesting and, as I said, very provocative topic. So, Dr. Ahuja, thank you very much for your time and for participating in our webinar. So, good luck with the presentation.
Thank you.
Can you hear
Gracias, Juan. Thank you, Sunil. Me olvidé, pero lo vamos a reparar, de decirle a todos los que están del otro lado que afortunadamente este webinar se realiza con la imprescindible colaboración de Altona, así que eso es justo y además también merece ser dicho. Ahora sí, dear Sunil, please.
Okay, Sunil, let's go. We're ready.
Okay, thank you. You're going to share this. You're going to show me the slides, right? Yeah. Okay, great. Thank you. You can hear me fine, Juan? You can hear me?
Excellent. All excellent, Sunil. We hear you and you see
Thank
you. Also.
Thank you. Well, thank you. I'm sorry that Andrea is not here. I've been to your institution, at least in the Children's Hospital, many years ago. I got to know Louisa and Andrea, some 25 years ago when they first came to my lab, and Louisa did a sabbatical with me along with Andrea for about a year, and Andrea came back several times. That was at the heyday when we had cloned CCR5, the HIV co-receptor, and identified polymorphisms in that gene. And a very large cohort of children from your hospital I have studied. In fact, we have the samples still over many years, and published those studies in Science and other journals. So I'm very appreciative of the long collaboration and friendship I've had with both Louisa and Andrea. So today I'm going to talk to you about a subject called immune resilience. And so this is but as I give you a bigger context, as in the introduction that was given, I run a Center for Personalized Medicine, and I'm a physician, and my goal is really to kind of practice this form of medicine called P4 medicine, where we can try to predict diseases, prevent diseases, personalize our therapy for our patients, and of course, have our patients participate in their healthcare. So this is the cover of Time magazine with a woman who you probably might know. Her name is Angelina Jolie. She's a very famous actress. And so the concept of prediction and prevention is very well illustrated by her, because she found out that she shared the BRCA1 mutation with her mother, who had died of breast cancer and ovarian cancer. And she elected to get a double mastectomy and then oophorectomy. So that really put the shine on to personalized medicine and prediction at that time. So, of course, I have spent a fair amount of my career in identifying genetic polymorphisms and other biomarkers, if you will, that will allow us to predict, prevent, and personalize the treatment of HIV and COVID. And I'll only share a little bit of that with you today. So along the way in this journey of taking care of patients with HIV many years ago, this is my second pandemic now. I started my career at the height of the HIV pandemic in New York City. But for example, when we took care of many of the children from your hospital in Argentina, it was very clear there were some children who did extremely well. We were also clear that there were children who were born to infected mothers who did not become infected. So we obviously knew there's variable resistance to acquiring infections. And in some cases, there's a big paradox of some people live longer and some people live shorter. In this case, there is a picture of a nun who developed COVID just before her 17th birthday, and she did not develop any illness. So obviously it wasn't just age. There must have been some other factor that protected her. She incidentally just died a few months ago, but not because of COVID. So I want to introduce the concept to you of salutogenesis, which is the health. So we spend a lifetime studying why people fall sick, but we spend less time trying to understand why do we remain healthy. Now, the reason why they are different is because the processes that cause health are often different from the processes that cause disease. But so I've spent the last, I'd say, a decade or more trying to understand really the process of health rather than what keeps us falling sick. So since the time we are born till the time we die, we are obviously have to interact with an environment, and that environment, like infection or trauma or allergens, they place an inflammatory stress on us. And then the concept I have is that we were born with some innate immune resilience, and that innate immune resilience wears away with time. But there are some people who have a capacity to preserve this immune resilience, and they live a longer and healthier life. So this whole idea, actually, some of it began when I read this editorial by Mark Davis, who some of you might know is a very famous immunologist. And he said that if you went to your physicians today and asked, you want an immune checkup, they wouldn't know what to do for you. If they ask for a cholesterol checkup, no problems. They'll be happy to do that for you. And although we use mice models extensively to understand the basis of human diseases, virology, etc., we really don't have been able to transfer that information to how do we take care of our patients. So what I'm going to show you today is the concept that there is immune resilience. It can be optimal or suboptimal or non-optimal, and we can quantify it. And because we can quantify it, it has relevance for prediction of diseases, a variety of diseases. This is the culmination of a very large body of work. So the title of this study just came out a few months ago in Nature Communication. It states immune resilience, despite inflammatory stress, promotes longevity and favorable health outcomes, including resistance to infection. So the most important word here is despite inflammatory stress. So the whole point is, just like we have emotional resilience, we'll never know whether we have emotional resilience unless we have someone emotionally harassing us. like our friends, our colleagues, or our family. So same way, we will not know how good our immune resilience is unless we have inflammatory stress. The idea being that there are people who, despite getting inflammatory stress, manage to preserve this trait that we call, which I'll define for you in a minute, called immune resilience. And if they manage to, despite inflammatory stress, preserve immune resilience, as I will show you in a few minutes, they have a unique trait of living longer, better health outcomes, and resistance to a variety of infections. This work of ours that was published was accompanied by an editorial or a blog by the NIH director. But the most important thing for me was that there was a comment on one of the, I don't know what's called, Twitter or X. The author is at the bottom. It says here, author resilience when it takes 2 years to get your paper published. So our paper went through multiple rounds of statistical reviews, etc. It took a really long time to get it past reviewers to understand the concept of immune resilience. So these are my colleagues who helped this work. The study was actually encompassing 5 international sites, and it was a very large body of work, which I'll describe. So as you know, since the time over the last century, we've had improving lifespans, and that lifespan is basically correlated with an improvement in public health, like improving our water, our sanitation, vaccines, etc. So, but as we live longer now, there is an improvement, there's an increasing antigenic load, because before, everything that happened to us was truncated over a period of time. Our ancestors didn't live beyond the age of 50, as you can see here, till the 1900s. Lifespan has really only begun to increase over the last 50 years or so. And so we really need to be able to have processes that manage this antigenic load. And that might promote health span and lifespan. So there's a consequence, of course, of living longer. The slide is not very clear here, I'm not sure why. But as we live longer, we have a risk of getting other diseases like inflammatory diseases, autoimmune diseases, etc. So this work actually, in many ways, began by this paper we had in New England Journal of Medicine, where we took people who were treated very early during HIV infection, within weeks of their infection. And what we found was that contrary to what we had expected, so what you see if you look on the upper left, this pointer doesn't work, unfortunately. So if you look to your upper left, is on the y-axis is the CD4 count, and the x-axis here is the month from EDI, stands for estimated date of infection. So you can see there's a vertical line here at 4, which means that 4 months from the time of infection. So we are managing to anchor the data right at the time of infection. There's actually an increase in the CD4 count, which actually peaks and then it comes down. So that means that initially there isn't a loss. There's actually an adaptive mechanism to actually improve the CD4 count. And then thereafter, there is this decrease in the CD4 count, which suggests some adaptive mechanisms there. We replicated this work in another paper in JAMA, Internal Medicine, showing the same thing, that early on during HIV, there's a rapid increase. So to do this work, we actually had to define what is the normal CD4 count in human populations. We took 14,000 HIV negative people and established the median CD4 count in human populations is about 800. And this will become important in a few minutes. So one can then imagine that there's obviously a trade-off of living longer. The trade-off is we have many more cycles of this inflammatory stress that leads to a greater burden of age-associated diseases. So when we get these cycles of infections or inflammation, sometimes we have a complete recovery and sometimes we don't have complete recovery. So if we have incomplete recovery, that would be a process that could lead to, for example, inflammatory aging that would predispose people to getting infections early. So the idea is quite simple, that we experience inflammatory stress. We can't get away from that. But there are 3 kinds of people. One, who are resilient. So they have less amount of senescent cells. I won't be able to show you all of that data. So there's another group who adapt to it, who have slightly more senescent cells on the right-hand side. And then those who are maladaptive, so they'll have a greater burden of these comorbidities or infections, etc. So what I'm going to show you is that actually immune resilience is a very quantifiable trait. It is sexually dimorphic. So women, as we have known for many years, are more immunocompetent than men are. They live longer lives. They have better vaccine responses. The yang of that is that they are greater risk of autoimmune diseases. So immune resilience is sexually dimorphic. It's quantifiable. So we've identified markers that you can use to measure it. And it is associated with superior immunity-dependent health outcomes. By that, I mean fighting viruses, for example, like COVID or HIV, as I will show you, or influenza, as I will show you. So the slides did not come out as clearly at your end, but I hope you can make out. So this is looking at the survival plots in people from the Framingham Heart Study. And the idea here is that on the upper two, so the y-axis over here is the proportion surviving, x-axis is the time since measurement. So we're looking at this data over nine and a half years, and we're stratifying the upper two lines in red and green are younger people. And you can see that over 10 years, younger people also have variable rates of dying, less so in females than in males. And then the bottom two, left side, the darker blue and light blue, are 67 to 92-year-old people. They also are obviously dying more faster, but again at a variable rate. And again, females die slower than men do. So next slide. So that was one paradigm, that is lifespan varies. The other paradigm, of course, is COVID. So, people associate COVID obviously with a disease that is found more commonly in older people. But here in the middle is an example of a person I knew well. He was the chief of ICU in Baltimore. He passed. Then there's another person who was 35 years old. He died. So obviously, age plays an important role, but independent of age, over and above age, there must be some factor that might predispose people to be susceptible or resistant to develop progressive COVID, regardless of age. So over and above the effects of age, there's a factor that plays a common role, whether you're young or you're old. And we actually had developed these concepts before the COVID pandemic. And when the COVID pandemic began, we applied these metrics, as I will show you, of immune resilience. And I monitored all our patients who came to our hospital in San Antonio, some 2,000 of them every day, to monitor their health status. And so we used these marker sets that I will show you to risk prognosticate. So we wanted to identify a common mechanism that, regardless if you're young or old, would predict their outcome. So we know this to be true because people have actually done the experiment. They took younger individuals, they inoculated them with SARS-CoV-2. This is a paper that was published in Nature Medicine, and they found that only 53% of these people became infected, which is to say there is the trait of you can get exposed to SARS-CoV-2 and yet you remain uninfected. And of those who did become infected, about 11% of them, you can see the bottom there, remained asymptomatic, and 89% developed mild to moderate infection. So I gave you the example of variable rates of disease progression, variable COVID, and you're well aware of this paradigm of women who was a very famous cohort in Nairobi, in the Maheno slum area, which have been highly exposed to HIV, and many of these women have remained HIV negative. And I'm going to today show you data from this group of women and show you how immune resilience plays a role here. We know, for example, when you take influenza, both in the natural infection or when you do challenge studies in younger adults, I'm going to show you some of that today, that about 1 in 3 people who, despite getting challenged with the virus, and sometimes up to 50%, remain asymptomatic, both during challenge studies as well as seasonal flu. And the question is, could someone's immune resilience be a protective factor there? So the last model system that I'm going to show you, so I've told you about lifespan, COVID, HIV, and then the last one over here was the issue of cancer. So as we know that there's always this big question, which came first? Did immunosuppression come first and then come cancer, or cancer came first and then immunosuppression? So as a model system for that, we used post-transplant skin cancer because after people get a transplantation, like a kidney transplant, we know they have a 100-fold excess risk of developing recurrent cutaneous squamous cell cancers, and we use this as a model system. So the idea then is that through life, aging, infections, or otherwise, we have this inflammatory stress, and it leads to a transient decline in immunocompetence and an increase in inflammation. So IC low, so that's immunocompetence, abbreviated low, and IF standing for inflammation high. So we develop this IC low, IF high status. But there are some individuals who are privileged who have the capacity to preserve this status of high immunocompetence and low inflammation when they experience inflammatory stress. And these individuals we call as having optimal immune resilience. Females are more likely to have it than males. This associates with longer lifespan, as I'll show you, asymptomatic COVID, asymptomatic influenza, sepsis survival, and resistance to HIV/AIDS. So this entire study is very simple. So as this abstract of our work says, some people remain healthier throughout life than others, but the underlying reasons are poorly understood. Here we hypothesize this advantage is attributable to optimal immune resilience, defined as the capacity to preserve and or broadly restore immune functions that promote disease resistance or immunocompetence and control inflammation in infectious diseases as well as other causes of inflammatory stress. So the key take-home point is there are two things combined together. You have to maintain high immunocompetence and concurrently maintain low inflammation. It's not absent of inflammation. Inflammation is absolutely vital. You have to have the right amount, at the right place, at the right time, and that's absolutely vital. So you can't escape inflammation, but it's a controlled inflammation. So the idea is that when you get insulted by the inflammatory stress, some people maintain this high immunocompetence and low inflammation status. And that duality, these two things together, is what we define as immune resilience. So again, this figure did not show up well, but now imagine on the left axis, on the upper left is IF, stands for inflammation. On the bottom left is IC, or immune competence. So when it says on, that means the inflammatory stress started and there is a burst of immune injury or inflammation. And then when the stress goes down, there's repair mechanisms happen. If you look at the bottom dotted line, there will be a decline concomitantly with inflammation and then back going up. Now, oops, so this did not work then. So my thing did not, oh, nope. So my, what do you call it? Animation is not working. Anyway, so basically the slide over here was to illustrate that we go through these multiple cycles and there'll be some people who basically remain resistant and the load remains low. So they maintain this high immunocompetence, low inflammation all the time, whereas the others become non-optimal. The cycles become much more, and then they have a higher amount of low immunocompetence and high inflammation status as age happens. So again, this will not show, unfortunately. Oh, well, okay. So what we did here was, the idea was we took these different model systems, the model system of aging, the model system of HIV risk factors, as you can see in the upper top there, the SARS-CoV-2 or COVID-19. We took the post-transplantation model in HIV. So increasing grades of inflammation. And the idea is that when we get exposed to these inflammatory insults, we maintain a concept of immune allostasis, an adaptation. Some people adapt well, like I said earlier, that would be optimal, that's on the left side, or it could go shift all the way to the right side, which would be non-optimal. So what you're not seeing here, because the animation is not working, is that the optimal is associated with these metrics, and it is more likely to be in females, which I can't show you here. So we'll have to just move ahead for the moment. So you can see in the upper left now over here. So in the upper left here, if you train your eyes to the upper left now, you will see that the optimal state is associated with being females, and IC high, IF low status. And these are individuals who, despite getting inflammation, they keep preserving that status over there. So if you look to your upper right, where it says resist IR degradation, we developed two metrics. So what you'll see the letters there, IAG1. I hope you can see it. It's not very clear. So that is immune health grade 1, which is one metric. And the other metric you'll see here says SAS1 high and MAS1 low. So SAS1 high and MAS1 low represents gene expression signatures or transcriptomic signatures that we developed. So SAS1 stands for survival associated signature, and MAS stands for mortality associated signature. So as you can imagine, if someone has got a high expression of a survival associated signature, it's good. And when you combine it with low amounts of mortality associated signature, that would be good. So that combination is another metric. And on the bottom right, these are the outcomes that we examined: lifespan, COVID, sepsis, HIV, etc. So we studied about, again, you can't see the writing very clearly. It's about, this study was 50,000 people and a variety of cohorts. We also studied monkeys and we also studied mice. But for today, I will only be talking about some human studies. So here, again, I apologize. On my screen, it looked fine, but obviously over here, when it comes across the internet, it's not very good. So bear with me. So on the upper left side, there is the CD8, CD4 profile, or the immune health grades is one metric. On the right-hand side is the transcriptomic profile, like I mentioned, SAS1, which is the survival associated signature 1, and they comprise immune competence genes, and MAS1 comprise inflammation genes, and they associate with mortality. Okay. So what we did was that based on the relative, here what we did was, as I mentioned to you earlier in our previous work, we identified that the median CD4 count in normal human populations is about 800 cells. And here we used the CD4 count of being normal, and we used a CD4-CD8 ratio, whether it's less than 1 or greater than 1, as a proxy for how much CD8s are. So grades 1 and 2 are good grades because they associate with restrained CD8 expansion, either with higher CD4 counts, which is the grade 1, or with lower CD4 counts, which is grade 2. Grades 3 and 4 aren't so good because they associate with CD8 expansion with higher CD4 counts, which is grade 3, or with lower CD4 counts, which is grade 4. Unfortunately, these animations are not working, but I wanted to show you that you cannot infer someone's grade by simply looking at their CD4 or CD8 count or their ratio. You need to actually create these grades in the schema like I've shown in the upper left to be able to create these grades. So here is about 3,000 people, actually it's 4,000 people, followed from the ages of 15 to greater than 90. So the y-axis is the immune health grade, and the x-axis is the age, and the color schema is on your right. And here we're looking at the balance of CD8, CD4, those immune health grades I mentioned to you a minute ago, blue color representing the best grade and black representing the worst grade. So as you can expect, blue is best, brown is number 2 is great, grade 3 is green, and black is grade 4. As we age, our capacity to preserve blue goes down. And then there's a greater expansion of people having grade 2 and grade 4. So for example, I belong to a grade 2, I don't belong to a grade 4, and I'm in my 60s. But about 5% of young adults, as you can see where it says the arrow on the upper left says susceptible, these are individuals who at a young age, between 15 to 29, actually developed a bad grade, which is more commonly found in older people. And similarly, where you see the word resistance and going to the arrow in the blue in the greater than 90 years of age, these individuals, despite being older, have preserved a good grade. We commonly study age as a categorical variable. We compare older people to younger people, but what this slide is showing you is that perhaps a more appropriate way, if you will, to study aging, if you will, as I'll show you in the subsequent slides, is to compare older and younger people according to the immune health grade. If we do that, then in that case, you might find that the immune traits of people who are older and blue are similar to the younger people who are blue. And similarly, the people who are black and older are similar to the younger people who are black and older. Or another way is you could give influenza vaccine to those younger people who are 15 to 29 years old, that 5% of young people who have it. You will find that they are more likely to have a poor influenza vaccine response, just as much as among the older population. We find that if we give a flu vaccine to the black group, older people, they too are more likely to have a poorer response. Now, what is not showing here is, I had a slide over here, it's not showing, the animation is not coming through here, is that across age span, more women preserve blue than men do. So that is not showing over here, but you can see it over here in the next slide. So in the next slide over here on the upper left, what I want to show you is that there'll be a sign on the top that says HIV negative, and the right side is HIV positive. And what I want to illustrate to you over here are a few concepts. That this change in grade that occurs with age is not likely to be due to chronological age. It is more likely to be the effect of the accumulation of inflammatory stress we experience during aging. So if you have a younger person who is experiencing a higher burden of inflammatory stress, they're more likely to have this even if they're 5 years old. So which is what I will show you in a minute. So here on the upper left, this is the Sardinia cohort of about 4,000 people. And at the bottom here, if you train your eye to the left, you'll see female and male. And you'll see that this is the overall cohort, not across age, but more females have got blue than men do, and more men have got green and black and brown. And in the previous slide, we went across age. So this is in the middle. After that is exactly the same slide I just showed you. So then where it says schisto means these are 5-year-old children who have got schistosomiasis. They're from Kenya. And at the bottom there, you see the word eggs. So we looked at the amount of egg they had in their urine. So these are schistosoma eggs. And what we are asking is that if the child has got more than 500 eggs, which means a high inflammatory burden, compared to say 1 to 99, which is less burden, what you'll see is that from controls, which means 0 eggs, all the way to greater than 500 eggs, there's a progressive decline in the amount of blue, which is to say this greater inflammatory stress reduced their capacity to preserve the good immune health grade. So nothing to do with age. It had everything to do with inflammatory stress. I will show you again in the next few slides. These are FSW, stands for female sex workers. So when you see the score there, behavioral activity score less than 0 means they had less sex activity, used more condoms, less sexually transmitted infections, they had more blue. As they went to greater than 0, that means they had a greater behavioral activity. The amount of blue went down and the amount of brown or green and black went up. And similarly, if you look to the far right, you will find that it says EIC, which is an early HIV infection cohort of about some 5,000 people. And SVC stands for, as you might know, there are some HIV positive people who manage to control their virus spontaneously. So the SVC stands for spontaneous virologic controllers, and LTNP stands for long-term non-progressors. So these are elite group of people who have HIV, and you can see that they have a much more amount of blue, these are all therapy naive people, and then say the people who don't have that trait. So less blue means they don't have the trait of controlling the virus. They got less blue. There's those who spontaneously control it. They got more blue and less black. So this changes, if you look at the bottom now, where it says antigenic load, inflammatory stress, low, moderate, and high. As we increase the stress levels, we are more likely to have a breakdown in this grade, which is tracking immune resilience. Again, this is not showing you everything that we studied here. So we monitored, I'm going to first show you our first paradigm of COVID, for example, where we took these grade systems that we developed and we monitored them daily in our patients for nearly 2.5 years now. We managed to, obviously, because we have such a large cohort of some 2,500 people, we can control them by age and sex. We can control them by their comorbidities, by the inflammatory responses, etc. We monitored them with their C-reactive protein, their IL-6. We measured a variety of outcomes from looking at whether they developed ARDS or other severe infections. So that again is not showing up because of the animation not being there. Hopefully I'll be able to illustrate those concepts with this over here. Excuse me for 1 second. So what I just showed you is on the upper left side. So this is a schema of a cohort, if you will, of people who don't have COVID, so before COVID. And the blue color represents the best grade, grade 2 represents the brown, grade 3 is in green, and black is the worst, which is grade 4. And you can see that with age, the amount of blue goes down. Now, let us assume we did a thought experiment. So a thought experiment would be, which we cannot do, is we just shower this cohort of people with SARS-CoV-2, which we cannot do. But you yourself would predict that if you did that experiment, you would find that who before COVID had the black color, and they should do worse compared to those people who are in the blue color. Unfortunately, this animation does not work, so I can't show that to you. This is very unfortunate. If I had known this would not work, I would have prepared a different set of slides. So basically, what the idea is that if you have got a grade 1 before COVID and you switch to a grade 2, so you'll do worse. Or if you went from a better grade 2 to a worse grade 2, you will do worse. Or if you had a better grade 4 and you went to a worse grade 4, you will do worse. And that's exactly what we found. And that tracked the inflammatory load and was highly predictive of outcomes independent of age. This is not going to show you well here. So again, this is the schema that we used on the upper top over here, basically to show you that about 80, this is on the upper, let's just bear with me here. On the upper top over here is group of people called 2C, which has got a CD4 count of less than 200, and 4C to your right, which has got a CD4 count of less than 200. So if you look at the cohort as the control cohort, which says SAIL, what you will find is that 0.1%, you can see the number there, and 0.3 of them have got grade 2C and 4C. However, during COVID, nearly 18% of the population presents with this very acute AIDS-like immunodeficiency, if you will, which is they present with a CD4 count of less than 200. So about 13% of people present with 2C, and about 6% of people present with grade 4C. So that's a very unique feature that is found very uniquely with COVID, which I'm sure you're aware of, but we could stratify them and metricize this progression. So we have now since then, so there's obviously the group of people, only 20% of the cohort preserves grade 1. In normal populations, depending on the age, of course, between 56 and 76% of the population has preserved grade 1. So, but you can see in the upper left over here, when I say the word elite, so the elite meaning that this is a group of people who had grade 1 before they had COVID, and they managed to preserve this grade during COVID across age span. That 21% of people, regardless of their age, they had all kinds of traits, which we immunophenotyped over 65 immune traits in them. As I mentioned, we looked at a variety of biomarkers you can see over here, neutralizing antibodies, cytokines, and a variety of gene expression assays. And we found that this group of people maintain more neutralizing antibodies. They have more marginal zone B cells, whereas the vulnerable group standing here for V represents a group of people, regardless of whether they're young or they're old, it does not matter. If they present with that grade 2C or 4C, which represents or tracking people with CD4 count of less than 200, and 18% of the population presented with that, these people have high viral loads, they don't produce much neutralizing antibodies, they have all the immune traits of a very activated immune status. So I'm just summarizing what we have found, and this is a summary slide. So if you look here in the middle over here is a slight brownish color where it says immunological resilience in AC COVID presentation. So this is their immune status at presentation. The bottom line was that there are a group of people who preserve grade 1. They've got a high immunocompetence, low inflammation status. These individuals, when we look back how they presented, they had actually, across their lifespan, and regardless of their age, they had a lower comorbidity burden. When we go forward in time, so during acute COVID, so during the first 30 days, when we look at their mortality, their hospitalization, the ARDS, severe complications, viral load, very minimal, all good status. And then we followed them for another 2.5 years. Beyond that, again, their rate of mortality is lower. On the other hand, there's this group of 18% of people who are present with this very acute immunodeficiency. When we look backwards in time, these are people who have had a greater comorbidity burden before they developed COVID. During COVID, they had the highest mortality risk after adjusting for age. So we can control for age, we can control for sex, we can control for 7 different biomarkers. Actually, we have now controlled for nearly 20 biomarkers. It is an independent determinant of outcomes. And when you go forward in time, they have a greater post-acute mortality and greater post-acute conditions than people who preserve their grades. So I'll just give you some examples of that. So this is an earlier paper that came out earlier before a Nature Communication paper with Jackie. So this is a cohort here. You can see blue color represents the best grade in patients with COVID. Lots of brown now. If you remember, there was lots of blue before, and it doesn't show up over here, but let's see if I can show this over here. So this will show you the paradigm. So if you can train your eyes for the moment over here at the bottom left, you will see that I have divided the cohort up into different age strata. So to your far left, there are 2 columns, and they say NH and H. So NH stands for not hospitalized, and H stands for hospitalized. So this group of people from 21 to 39, 40 to 49, 50 to 59, 60 to 69, and greater than 70 years of age. So even though the hospitalization rates went up with age, as you would expect, I would like you to look up to the top now. What you will notice is that in every age bracket, the first column, which stands for NH, has got more blue. The second column, which stands for hospitalized, has got less blue. So more blue, less blue, more, less across age. So it doesn't matter if someone is an older age bracket or a younger age bracket. It is more likely that if people preserve their blue grade and have less black and green, black grade, they are more likely to have been not hospitalized. On the right side is actually telling us, if you look to your the bottom is again the y-axis, the bottom x-axis is age, and the y-axis is the percent hospitalized. The 2 dots that you see on the top, the brown and black, represents the probability of being hospitalized if someone presented with grade 2C or 4C. And if you train your eyes on the blue line, what you will find is that 80-year-old people who came in with grade 1 had a lesser likelihood of being hospitalized than a 20-year-old person who came in with grade 2C or 4C. So these are very predictive of outcomes. Again, my animation is hiding the screen, but you can see this is the Kaplan-Meier curve. And if you just train, y-axis is 30-day all-cause mortality. The y-axis represents people who had died, percent alive. And if you just train your eyes on the 2 curves that are coming furthest down to your left, they represents grade 2C and 4C. They have a 10 to 13-fold excess hazard of dying rapidly after adjusting for age. I'll skip this slide in interest of time here. So this, again, the slides are not going to show up completely, but what I want to illustrate over here is on the y-axis, on the x-axis is age, on the y-axis is, for example, 2 parameters here, NK cells and ferritin. What you'll see is that the blue line, which represents IgG1, across age, those people who preserve grade 1, they had higher amount of NK cells, and at the bottom over here, ferritin, which is the biomarker we can use to see lung injury, they got lower levels of this. If this animation had been removed, you would have seen that the people who had IgG1 had maintained across age low IL-6, low CRP, more B cells, and less amounts of D-dimer. So this is what we did every day. Dr. Fauci was my mentor at the NIH, and he trained me when I was there. And so this is how we would do our rounds every day. We would monitor our patients with these grades. And if they came in with 2C, 4C in the very early part of the pandemic in early 2000 itself, and my recommendations, we had started to give these therapies for anti-IL-6 monoclonal antibodies well before it became a standard of practice at our nationally or internationally. So these are some other examples to show you that this is a cohort of older people who are in the nursing home, who we are monitoring their neutralizing antibodies over 400 days. And we are, if you look at the blue line, that represents people who are older and who came in with grade 1, which is blue color, and black is at the bottom. And this is on the y-axis is their percent inhibition. The higher the level, the more the neutralizing antibody. And you can see that over 400 days, despite being older, those who had the black grade maintained lower antibody levels. It initially went up and then comes down, but it's always less than the blue. So a variety of traits are associated with better outcomes as they maintain this good grade. I won't belabor the point, but you can measure cytokines and you can measure a variety of immune traits, but those who preserve this best grade are what we call as being immune resilient. So I'll skip this in the interest of time. So I mentioned to you that it's common practice to study people by age, but that this could be a misnomer, because I mentioned to you here, so here's a slide that did show up. So you can see here on the right-hand side by sex, more females have it than men do. But the idea is then that we could be confounded if we just compare older versus younger people. So to define the immune traits, what we did was we categorized people according to their grade and their age. And then we immunophenotyped 4,000 people and identified 4 classes of immune traits. So the 4 classes of immune traits are over here. So one class of immune traits are those that only associate with their grade, not with age. There were 13 of such, which is under the word IR, the 13 such traits, like CD127 null, CD8 bright cells is a very prominent trait associated with only immune resilience or the health grades, not with age. Then there are immune traits that will only change with age and not with their grades. Then there's another set of traits that will change with both the health grades as well as the age, and another set of 30 traits that are associated with neither. So here, for example, is I'll just share one signature here, which is signature 1. The blue color compares the levels of CD127 null CD8 bright cells among younger and older people who are grade 1 in blue. No difference. Brown color, if you see less than 40, greater than 70, among people who are grade 2, no difference. But if you compare it by grade, at any age, you correct it for age or you look at it in young people or older people, it doesn't matter, that trait changes. So this is a way to prevent confounding when we begin to study our patients. So the next is this biological system that I mentioned to you of studying women who are sex workers, for example. And this is on the upper left. On the upper, if you look at, I'd just like you to look at a cohort that we studied for. They came in, we measured the immune health grade at baseline, and they were followed prospectively for over 10 years. So we're going to ask the question, does someone's baseline immune health grade predict their future HIV risk? I'd like you to train your eyes on the word BAS, where it says behavioral activity score. So as the behavioral activity score goes up, you'll see there are 2 sets of columns. The first is in black, which is the HIV proportion of women who became HIV positive in each of these women who have a behavioral score from -4 to 3+. On the upper are the colors for the immune grades. And as you can see that as the behavioral activity score goes up, the HIV acquisition rates go up and the amount of blue goes down, and there's more green, black, and less amount of brown and more brown over time. What we showed was that so you can see that even when women have this high behavioral risk activity, high HIV acquisition, some women preserve blue. Most of the HIV infection, virtually all of it actually, occurred in women who were green or black. So, which is to say that is a risk factor over and above their HIV exposure risk that predicted the risk of acquiring HIV. Obviously, if you train your eyes now to your bottom left, this is a group of women who we followed for up to 10 years. Every 2 years, we measured the immune health grade. And what you can see is we obviously told them to reduce their behavioral sex activity. So you can see to the right, it says 0 to 2 and 8 to 10 years, two plots. You can see that their score went down, and over 10 years, their green and black went away, and they reconstituted blue. So you can induce it, and it's reversible if you know the exact cause of their inflammatory stress. If you train your eye to the right, these are the same health grades in 5,000 HIV positive people who are therapy naive. These are Kaplan-Meier curves followed for 25 years. And we are asking the proportion who are AIDS-free. Those who had grade 1, it's on the top Kaplan-Meier curve. They live longer. Grade 4 to your far left, and grades 2 and 3 are in the middle. So how do you do lifespan? So what we did was we developed these scores that I told you called Mortality Associate Signature 1 and Survival Associate Signature 1. So again, unfortunately, it didn't show, but basically what happened was that we had screened or done a transcriptomic profiling of our COVID positive patients. We identified on the upper left where you see in the bottom on your left side is green, which means that these gene signatures, the higher their expression, the more their survival. That is why if you look to your very bottom, you'll see the word mortality and an arrow going leftward and an arrow going rightwards. So higher expression of those gene signatures in green, the longer the patient lived. And then on the top are inflammation genes. So these bottom green correspond to immunocompetence genes, signatures. And the brown on the ones on the top represent those that relate to inflammation. And so higher those expression of those gene signatures, like the type 1 interferon, you can see nice and bright and red. So that signature, higher expression associates with mortality. So we identified a set of signatures that are associated with mortality, a set of signatures that are associated with survival. So I asked my group to take the same signatures and apply it to the Framingham cohort. And what happened was, like the red on the top, you see that signature only associates with mortality in the COVID cohort. What you can't see here because of the animation being covering it, that it did not associate, obviously it's not a COVID cohort. In the Framingham, it did not associate with mortality. But like the green signature that says defense response to gram-positive, it associated with increased mortality in the COVID cohort, as well as during natural aging, it associated with a shorter lifespan in the Framingham cohort. If you look to your bottom left, there are three signatures there: T-cell co-stimulation, immune response, and T-cell receptor signaling pathway. And that signature, higher expression associated with survival in the COVID cohort and also associated with longer lifespan in the Framingham cohort. So basically we identified these signatures that are associated with survival and with mortality. So if you combine them together, you get more information. So if you look to your upper right now, so what you'll find is that there are four colors here: blue, gray, orangish, and then this yellow or brownish. So blue is the best. So they've got high immunocompetence genes, or SAS1, and low MAS1, low inflammation genes. So you would expect that these group of people should live the longest. And so if you look to your left, that's a Framingham heart study. You can see over 10 years, the lifespan of the one in blue is the best. The yellow is the worst. So they've got low SAS1, low immunocompetence, and high inflammation. They're the worst. And you can see they progress much faster. And the ones who are either bad, they have an intermediate rate of lifespan. So again, showing you that. And this immune health grade, this high-low trait is overrepresented in the people who have got immune health grade 1. So that's shown over here. So that if people have got immune health grade 1, they're also more likely. This is the cohort of lupus, and this is the cohort on the right with COVID. If you train your eye where it says IHG1 on the left side in lupus women, whether they're younger or older, it doesn't matter. If they've got grade 1, they've got more blue, so they're more likely to have this high SAS, low mass trait. In interest of time, I only have about 5 minutes left. We basically looked at an influenza infection cohort. And so here you'll see on the left-hand side, there's a group of people who we studied before they got an influenza. And you can see that they're mostly all blue. These are younger adults, less than the age of 50. They were all given a cup to say when you spit, when you get your first infection. And you come back, and they were measured to see if they had the virus, influenza or other rhinovirus, etc. And you can see that on day 0, when they had their first infection, they all turned yellow. So that same signature that associates with mortality gets turned on. And over time, the signature yellow reconstitutes down and blue gets recovered. What you cannot see on the right side is we took the guys who are blue before they got infected and followed them over time. About 25% of them did not recover their blue, which is to say they had recovered from their infection. In springtime, they were fine, they were walking around okay. But when we probed these guys who had this survival signature blue before they got their flu, 25% failed to recover back blue, which is to say you're left with some level of immune suppression or decrease in immune competence, if you will, after the flu. So this again will not show, but basically what we did was we took the gene expression data sets from young adults who were given influenza intranasally. And on the left side, you can see T1 and T2. So there are 50% of the people after intranasal inoculation remain asymptomatic. The other 50% became symptomatic. When we compare the gene expression when the symptomatic people are having their most symptoms and compare that gene expression to people who are asymptomatic, we found that those who remained asymptomatic preserved more blue. That is, they resisted a degradation of that yellow color I just showed you that happened during natural infection. I'll skip this in interest of time. The last thing I want to show you is that if we take another model of immunosuppression, which is in this case renal transplant people, so we use this model because people get recurrent skin cancers. So we asked, at the time they got their first skin cancer, if depending on their grade, what was the rate of getting their second cancer? So what we find is that those who, if you look at, so the y-axis is the proportion remaining free of this cancer, x-axis is the time from their first cancer to their second cancer. So here you can see that those who had grade 1 resisted getting cancer, those who had the worst grades had a higher rate of cancers. So what I finished telling you is that this is a quantifiable trait, immune resilience. We can metricize it, we can monitor in our populations, that I use these 4 different model systems to show that to you, that it is highly protective across a variety, so it's antigen-free, that there is a lot of immune heterogeneity in the human populations. So if you look, for example, to your far right, those are 40 to 50-year-old people. They've got yellow. Yes, 81 to 92-year-old people have also got a lot of yellow. But the point is that if I'm 40 to 50 years old and if I've got a mortality signature, I really want to like it, right? So, and then when we do these immune studies in our patients or in our participants, we're likely to be confounded because we're comparing 4 groups together. We collapse them together. And all we do is compare across age, which I don't think is a very fair way of looking at immune profiles. This is my last slide. So basically, in data that I'm not showing you, basically we found that this trait is highly associated with a very important transcription factor that regulates T cell fate. So TCF7, the higher, so if you look to your far right bottom, you will find that what we've done is taken age groups of 66 to 92. On the left side is 40 to 65. O stands for octiles. So increasing levels of TCF7, as we increase it, the capacity to preserve blue goes up, whether you're young or you're old. So I'm going to leave you with 3 ideas. There are 3 challenges in my mind. One is that I think there are 2 distinct forms of immunosenescence. One relates to antigen or inflammatory stress and it's independent of age. The other, I think, is directly attributable to chronological age. The problem in the field has been that these 2 forms have been conflated and they lead to confounding. The second I mentioned to you is the heterogeneity in human studies. It's vast and we need to find ways to control it. The third challenge is how do we reconstitute immune resilience, which is a very big challenge. So these are my authors. I don't have a slide showing their faces. This work was funded by many organizations, and I think I've time, and I thank you for your attention. I can't hear you. You're muted. You're muted. You're muted.
Yes. Now you can hear me?
Now I can hear you.
Okay, thank you very much, Sunil.
Yes.
Thank you very much. Great talk. Congratulations. I want to congratulate you because you're a very resilient author, as someone said, but also a very resilient speaker, because you managed very well to do the talk with the problems that that you have. So I don't know if
I'm sorry about that.
because you study resilience so much. Don't worry, it was very clear. I have a lot of questions, and I have questions from people coming from WhatsApp. And let me check. Okay, so I'm going to start from where. Okay, let's start with this because under your under the idea of immune resilience is this idea of that antigenic stimulation and leads to inflammation and to at the end lower immune competence with time, repeated antigen stimulation. But this idea that this is going against immune competence is like in contradiction with the idea of immunological memory, because with many antigen stimulations during life, you became more immunocompetent because of the memory. How do you deal? How do you explain this? It seems to be like a contradiction.
This is an antigen independent process. So there's going to be, this is not related to, no, I have to take it back. Those people who maintain this trait will have greater immune memory. But if they lose this trait, they will lose this process of maintaining memory. But this process is more directly related to an innate process because it is triggered immediately. So because we can inoculate someone in the nose, and within 40 hours we can see these changes occurring, and we can see there was a wonderful slide that I can't show you, but there are people who adapt, who become more blue during that short period of time. So that cannot be adaptive. And these are 20-year-old people who have obviously received vaccines and had flu beforehand. So I think that we innately had a process that allowed us to live longer, and that had to have happened across age span, especially evolutionarily, because if we didn't have it, women would not have gone through childbirth necessarily, cannot go through childbirth with a lower immunocompetence. So this is not an adaptive mechanism. This is a more of an innate memory. So it's like more for a trained immunity rather than for an adaptive immunity. So that is my view here. We I cannot show you the data today, but we have followed these. If I were to follow you through your COVID vaccination history, I would be able to show you that on the day of your primary vaccination, 7 days later, if you were blue before, or a group of people were all blue before, about 50% will not be blue on day 7. On day 21, just before their second vaccine, most of them are reconstituted back to 80%. They got their second shot, and now nearly 75% have gone from blue to yellow. So what I'm trying to get to you is this: we are experiencing every single day some form of antigenic stimulation, and we are going through these cycles. Some of us degrade, some of us and stay degraded, some of us reconstitute, and some of us never degrade. So there's immune resilient, there is an immune reconstitutor, and there's an immune degrader. The guy who becomes degraded, so 6 months later, we will find that they've got, with other signatures, they track greater inflammatory aging, so more agile 6, more inflammatory aging signatures, more senescent cell signatures. So I think this is a innate process. It's not an adaptive process. But those who preserve it maintain greater immunological memory. That's the whole point.
Okay, thank you. This took me to another question that you were talking about. Did someone in the chat? Yeah, someone ask you if immune resilience is more related to innate immunity than to adaptive immunity traits.
Which I just answered here.
Yeah,
which
you
I just
already
answered, right? Yes.
Yes.
So somebody asked me this
I
question
was wondering
about
let me add something. But it was surprising for me that antibodies are not associated with any of your studies. Are not
Sure.
immune traits associated with.
Sure. No.
Oh,
I showed you the data. Yeah, they are. I showed you the data from the vaccine. I didn't show you the vaccine cohort data, but we had a data set. It was hidden behind one of the slides. We developed a 1600-person COVID cohort, and we showed that those people who had grade 1 preserved higher levels of antibody durability over time. So that data is there. And then when we go in and probe other COVID or influenza data sets, the immune traits like B cell traits or plasma blasts, they are higher in people who have got grade 1 than those who have got worse grades.
Okay, let's go to another question from the public. Okay. Yes, someone want to ask you if maybe what variables should we take into account in addition to age or clinical health condition for applying the flu vaccine? So maybe the question is, do you think that we have to, so in the immune health grade could be a better predictor for the people that we have to choose for the vaccination than age?
Well, I mean, when we give our vaccines to people, more than 50% of older people, and
se cortó.
Sí, Juan, pareciera que se perdió la conexión con su. Vamos a esperarlo un ratito.
Se cortó la conexión con Sunil.
Sí.
Okay.
Vamos a esperarlo. Sí, gracias. Bueno, hemos tenido también en nuestras redes sociales un justo y hermoso impacto de la charla, así que estamos muy contentos. A ver allí, en cuanto lo vea Sunil, si se puede reconectar, lo hacemos ingresar para este último tramo de la charla, que como ustedes bien saben, no está de más recordarlo, va a quedar grabada en nuestras redes sociales, así que bueno, tenemos también esa ventaja. Todavía no lo tenemos, Juan, ¿no? Vamos a crear un poquito. Volvemos a agradecer al Tona que estuvo con nosotros, está en nuestras charlas siempre para que este tipo de encuentros y de diálogos se pueda realizar sin inconveniente.
Bueno, no sé si vos lo tenés en WhatsApp a Sunil para mandar algo.
No lo tenemos. Si te parece, no sé qué dice de él, por comunicación privada, pero si te parece, cerramos. Lo bueno es que la presentación de Sunil se realizó sin trastornos y fue muy provechosa.
Bueno,
si te parece...
Un montón de preguntas, ¿sí?
Sí. Bueno, cerramos, Juan, entonces, con unas palabras tuyas, ¿te parece?
Perfecto, bueno, les mandaremos el agradecimiento por mensaje. Muy bien. Muchas gracias, Diego. Y bueno,
gracias, Juan. Entonces, esto que siempre decimos en esta serie de encuentros, el agradecimiento por, bueno, atención, Sunil.
Volvió. It's back.
Entonces, te agradecemos.
Okay. Sunil, can you hear me?
Yes, I can.
Okay, we have another problem. Well, we were asking you if you think that some of the metrics that you show, probably the immune health grade, would be a better predictor for the response or the utility of the flu vaccine than age only, or maybe both.
We definitely can't exclude age. I mean, if you measure some of these markers like we do, we've examined over 100,000 gene arrays. 2 years of age among young people makes a difference. Age is so sensitive a marker, okay, a change. So all these vaccine studies are largely confounded by conflation. So I don't have a great data set other than the one we have for our COVID.
Okay.
So if we wanted to identify the correlates, I would prefer if we go forward by dividing up our vaccine cohorts or our cancer cohorts or whichever cohorts we study into groups, these 4 categories, and then go ask among them, where is the variability lying? So that is my experience is that, at least for me, like when we did our COVID cohort studies, that if you were in grade 4, the likelihood that you're going to fail your vaccine is very high. This is even among 20-year-old people. I'm talking about COVID now. I don't have a very large, most of these vaccine cohorts, if you look at them, they're 30, 40, 50 people cohorts. I mean, 60 people cohorts. We studied a 1,600-person cohort. And so the durability of the antibodies remain much higher. I just showed you among natural infection 70-year-old veterans in a nursing home, those who had grade 1 maintained across 400 days higher antibody response than people who had a worse grade. So if I extrapolate that, I see no reason why not. So in fact, there are colleagues of mine who had grade 4. They were terrified during the COVID time. And there are some who still monitor their grade every day and they wear masks because they are grade 4. So my entire hospital staff knows their grades and they monitor themselves all the time.
Okay. So this leads us to another similar question that is so in which clinical condition or what clinical situation do you think these metrics are going to be useful for patients other than the flu vaccine and then the one that we talked just?
Well, I mean, I think they would be useful for any disease condition where you care about someone's immune health, right? So, for example, if you had a person who is on, I give you the example for, say, for grade 4, or you could take HIV, immune reconstitution. If someone has not reconstituted back to grade blue, their immune health is very different and they have a more pro-inflammatory status than the other. We don't monitor it in our patients anymore. We just monitor their viral load, see if they have a blip, and look at their CD4 count. But that is a very imperfect marker. If you were going to follow a cancer patient going forward, or some of my patients who have got chronic osteomyelitis or other diseases, we monitor these grades all the time in them and see if they, what is the change occurring in them. At least for me, in my practice, I use this all the time now. A lot of my colleagues now, because of... Yeah.
And it changes a therapeutical decision?
It may not necessarily change therapy decision, but it definitely changes how well I continue to follow my patients. So post-trauma sepsis, I would follow them much more closely if they were walking out of my hospital with a worse grade. So I may be paying much more attention to them than I would have otherwise. I would highly recommend them, wear masks, do other preventive measures and things of that nature.
And do you think it would be in the balance of cost and benefit? It was something that everybody should measure. Maybe CD8 and CD4 is not very costly, but the other metrics might be.
CD4, CD8 is $67 it cost me. So at least in America, we measure it.
Yeah.
$67. So we monitor very frequently in our sepsis patients, our COVID patients. We did that. They've expanded that work to our cancer patients. So, so we are obviously blessed. We come from a country where we can, measure as part of routine care. So it does matter.
Okay. Let me check if I have something more. Also, this question is very interesting. Do you think there is an association of the immune resilient with some social habits or conditions, or maybe behaviors?
Well, one behavior I showed you, which was sex behavior, right?
Yeah.
So
Nairobi.
I will not publish this, but we actually have a cohort. It's on the online. We downloaded it. They had good metrics of oral sex and intercourse. Okay, so remember, so as the amount of oral sex and intercourse went up with multiple partners, it's not the same partner. If you're monogamous, it's okay. But if you have, and you can obviously, doesn't matter if you have the sex frequency. It is when you have multiple partners, because then what you're doing is you're alloimmunizing yourself. You're having different exposure to semen or to blood. So that's social behavior. We have looked at some data sets with socioeconomic status or with stress, where a father and mother are going through a divorce and the child is there, and that kind of stress. There is we don't have a very large cohort, but there's a paper in PNAS by Stephen Cole, which showed that this kind of stress leads to changes in their CD4, CD8. He didn't measure it by our grades, but if you were to look at it, it would basically look as though they're developed grade 4. So our problem is we can't do everything. A lot of our work is based on publicly available data and how well it's been well phenotyped. So yes, I do think that social stress plays a role. I wish I could study it more, but I would need a larger cohort for that.
Okay. And just to finish, Sunil, how do you think this field is going on? Which would be the next to do on what are you trying to see right now?
So I think we need to, as I said, the issue of heterogeneity in human populations. I don't know what, when you do your clinical studies at the hospitals, we don't take these into account. So I definitely think we need to control it. I think the aging field is largely confounded because of it, because all they do is compare younger and older people. So I think that's a misnomer over there. And I think the idea should be salutogenesis. What keeps us healthy, not what makes us sick. They're not the same things.
Okay. Well, Sunil, thank you very much. I think we're on time. So thank you for the talk. I don't know if you want to say something.
Muchas gracias, Sunil. Muchas gracias, Juan, a todos los secretarios que estuvieron del otro lado. Muchas gracias a Antonio, que nos acompaña como siempre. Un placer de compartir conocimientos con quienes lo necesitan. Nada más hermoso que eso para la Sociedad Argentina de Biología. Tengan todos muy buenas tardes.
Muchas gracias. Goodbye.
Gracias.